The overall goal of this procedure is to quantitatively assess the manual dexterity of macaque monkeys. This is accomplished by first transferring the monkey to a primate chair without stress to the animal. The next step of the procedure is to measure daily the manual performance for each hand in a reach and grasp task in which the monkey has to collect 50 small objects using the precision grip. After that, 3 other manual dexterity tasks are performed to complement the behavioral assessment. The final step of the procedure is to analyze the performance based on the video sequences collected during the 4 tests. Ultimately, results can be obtained that show the motor deficits following a lesion of the central nervous system and the extent of the subsequent functional recovery through the time course of changes in the behavioral parameters derived from the quantitative analysis.
The main advantage of this technique over existing method, such as clinical scoring, which is, by the way, subjected to large inter-rater variability, is that it is truly quantitative and reliable, with moderate inter-individual variability.
This method can help answer key questions in the behavioral neuroscience field, such as mechanisms underlying plastic reorganization of the central nervous system following a lesion or a neurodegenerative disease.
The implications of this technique extend toward therapy promoting neural regeneration and/or strategies of substitution after lesion of the central nervous system, because the non-human primate is often a mandatory step before clinical application of a therapeutic strategy initially developed in rodents.
The non-human primate is pertinent